He aha te ahau wāru mai, e tiu he rā ki nāna ia mai te pūputara ki uta, e tiki nā atu au, te kōtu, ko ia te pau whakairo ka tū ki Waitemata, i oku wairangi e, te ei wā mauri ora ki a tātou katoa, e hui mai nei i raro i tūnui o tēnei whare ke te mihi. Ke te mihi atu ki a koutou e tai mai nei mo tēnei kaupapa whakahirahira, ki te whakanuia i tō tātou rangatira, a Viji i tēnei wā, nō reira ki a koutou katoa, nā whānau whānui, ngā ahurangi e tai mai nei i tēnei wā, ki te tautoko i ai i tēnei wā, kei te mihi atu ki a koutou, he mihi mahana ki a koutou katoa. He maumahara hoki i rātou mā kua whetūrangi ki tū o te ārai, moi moi rā, moi moi rā, moi moi rā. O te rā koutou katoa, ngā akonga nui e tai mai nei i tēnei wā, ki a koutou katoa, kōkua te ngākau ki te kiti a koutou i tēnei wā, ki te tautoko mārika i tēnei mininga i tēnei wā, i rungi te rangi Māori e o tō tātou matunu i te rangi kei te mihi. O te rā ki tō tātou manukura tuarua, ki a koe Jan, he mihi kawana ki a koe. O te rā mō tēnei kaupapa, ki a koe hoki Michael, mai te tari o te Ihunuku Māori, e mihi kawana ki a koe, e tai mai nei ki te tautoko i tēnei rangatira i konei, he mihi kawana ki a koe. O te rā, ki ngā ahorangi nui e tai mai nei i tēnei wā mo tēnei kaupapa, he mihi mahana ki a koutou katoa. As the kaiarihi for the Faculty of Science, warmly welcome you all to this very auspicious occasion for the professorship of Viji tēnei wā. Homai te paki paki. I have to say, when I look out there into the lecture room, Viji, this is the most youngest audience that I've seen at such professorial type of gatherings. Not saying anything, but they're usually in the 50-year age bracket, but having so many young ones here just shows how much of an impact that you have as well to the youth, but also to the youth adjacent that have arrived here as well. Very nice words there. Welcome you, each and every one of you, into this beautiful place. The traditional warm welcome to each and every one of you, particularly on these lands and waters of Ngāti Whātua Ōrākei. And I also acknowledge here we have our Ihunuku Māori deputy from Te Tari of the PVC Māori office, who would usually take this role in welcoming you all here before as the first kaiarihi for the Faculty of Science, Michael Seidman, but also all the professors that have gathered here as well. And also I know that we have Jan here, who is our manukura as well, who's stepping in for Sarah Young, who cannot be here at this time, to share a warm welcome and say some kind words, celebratory words about you, Viji, as well. Given that there's such youth and vibrance in here, usually what we do in these warm greetings and welcomes, we do a waiata. We do a song as well. So what the heck? We're going to do that as well, whānau. And you all know it. Te aroha it is. And so we're all going to stand and we're going to go through te aroha together. E tū tātou. Here we go, whānau. Me tīmata tātou. Te aroha, te whakapono, me te rangi Māori e, tātou tātou e, anō. Te aroha, te whakapono, me te rangi Māori e, tātou tātou e. Me noho tātou. Ka pai koutou katoa. You did really, really well there. Nā mihi ki a koutou katoa, nā mihi mahana ki a koe, I te rangatira o Kerala, nā mihi kawana ki a koe I tēnei wā, mo tēnei Ka ike koe ki te taumata I tēnei wā nami kawana ki a koe. O tira ki tō whānau I taimai nei ki te tautoki a koe. Tēnā koutou, ka hoa te rākau ki a koe te.
Ina mana, ina rio, I rao rangatira mā, tēnā koutou, tēnā koutou, tēnā koutou katawa. Kia ora koutou, I'm Jan Eldridge, the Deputy Dean here at the Faculty of Science at Waipapa Tamata Rao, the University of Auckland. It's a real privilege to welcome you all this evening to our university and to the inaugural lecture of Professor Viji Sarojini. Professor Viji is a peptide chemist whose research explores the fundamental properties and wide-ranging applications of peptides. Her work has revealed their potential not only as human therapeutics, but also in protecting crops and enhancing the sensory qualities of food. Viji's journey began in Kerala, Viji's journey began in Kerala, a lush and vibrant state in the southwest of India. After completing her schooling and undergraduate studies in her hometown of Mavelikara, she moved north to Varanasi, where she earned a Master of Science in Medicinal Chemistry from Banaras Hindu University. Her doctoral research was carried out jointly at the Indian Institute of Science in Bangalore and BHU, and was followed by postdoctoral appointments at Linköping University in Sweden, the University of Leeds in the UK, and Texas A&M University in the United States. In 2004, Viji made the move to Aotearoa, and in 2005 she joined the Plant and Food Research, where she worked on developing bioactive peptide solutions to tackle bacterial diseases in plants. The following year, she began her academic career here at the University of Auckland. It's a pleasure to celebrate her achievements and contributions with you all tonight. Please join me in welcoming Professor Viji Sarojini to the lectern.
It's great to see a lot of people here. Thank you all very much for coming, and that in itself is an honor. So the next hour is intended to be a little bit of fun, enjoyable, something interesting for everybody in the audience, scientists and non-scientists alike. So I don't want to bore anybody, so I would say at the start, please feel free to laugh whenever you need to. In fact, laugh more often than not. Thank you very much. And so while I was preparing my lecture, I played around with different titles and finally settled on, just getting the laser pointers, just settled on this short one, which only captures one aspect of my research. There are other aspects to my research, and there are also other aspects to the academic journey, as you saw in the abstract, and so I would like to share some of those with you today. As you heard, I was born in Mumbai. Many people might know Mumbai, but we are from this small state, southwest state in Kerala, beautiful state, and it's actually, the landscape is very similar to New Zealand, I would say. Lots of beaches and mountains and so on. So this is my mother's hometown, Mavelikara. That's where I went to school, grade 1 to 4, primary school, and grade 5 to 10, higher secondary school. And after grade 10, we go to do grades 11 and 12 in a college. And at that time, it was called pre-degree. So I did pre-degree in my local college, Bishop Moore College, Mavelikara. I went on to do BSc right there. And then, as you heard, I went to do MSc, Banaras Hindu University, PhD. Most of the work was done at Indian Institute of Science, Bangalore. First postdoc was in Linköping. So you will hear a little bit more detail of these different aspects of my journey as we proceed. But before we proceed, let's look at some beautiful pictures of Kerala. I promise you, I'm not getting paid by the Kerala tourism to show these pictures here. But I thought I should show this, and I have not seen any of these places, to be honest, just one of the places. Because being a student back there, you don't have much money to travel, so this is what I'm going to do during my sabbatical, perhaps. So houseboats in the backwaters of Kerala, and coconut is an important regional cash crop. Every single household has their own coconut farm, or at least some trees. We never purchase coconut, and coconut is in every single dish, spicy dish, curry, sweet dish, you name it. And coconut oil, well, there was a lot of controversy when we were growing up. We all started not using coconut oil because somebody said it's bad for the heart. Now they say it's good for the heart. So now we are cooking in coconut oil. Western Ghats from the south to the north. So one side is the Arabian Sea, the west, and this is the Western Ghats on the east. Onam is the biggest festival, the harvest festival in Kerala. So that falls between the month, middle of August to middle of September. And this is a major thing, the snake boat race in Kerala. And this is the feast, 26 dishes or more than 26 dishes. And what happens after the feast is that the men go and start drinking. And now here, rich history, as you know, India has. And this is the rich history of Kerala. Lots of wealthy temples, churches, mosques, synagogues, you name it. So that's Kerala. And here is the family. This is my great-grandmother. You see the umbrella she's holding. That's actually, this is a photo maybe 80 years old, maybe more. This is an umbrella which is made of the palm leaves of the coconut tree. These are my maternal grandparents. I have never seen my paternal grandparents. They all passed away before we were born. My grandfather was working in Myanmar, Burma at that time it was called, a British school. And my mother was actually born there. These are my parents, my father and mother. They were simple people, but they were calm, but they were also firm when they needed to be. You'll hear some of that later on. So these are us. We are three, my eldest brother, my elder sister, and this is me, the youngest. My brother is no more, so we are two now. And so these are the names. And we are from a matrilineal system, meaning women hold power in the family. So if you look at the names, if you did not know, you will not know these are brother and sisters, because daughters took the surname of the mother and sons took the surname of the father. So this is one line which we were told every single day. I've given the English translation there. Vidya essentially means knowledge. Dhanam means wealth. And what this actually means is that there is no wealth superior to knowledge. And this is what our grandmother told us every single day for obvious reasons. First of all, it cannot be stolen. Secondly, it grows as you spend it, which is what we do in a university, I believe, and so on. So my grandmother, maternal grandmother, she played a significant role in bringing us up and also ensuring that we go into higher education and have progressive lives, together with my parents. So here you go. So this is my sister and myself. This was a function in one of the annual day functions in our primary school. We were not holding the smartphones at that time. This is just an image I generated from Microsoft. It was some teachers were retiring and we were singing, and it was all going fine. And then what happened, there was a small mistake in one of the lines. It really did not matter. Nobody noticed it. So you just keep singing. That's what my sister did. But what do? I couldn't tolerate. See, I turned to my sister. Everybody is sitting on stage. I turned to her with a very disapproving body language and facial expression. I'm looking at her. How can you do this? It's my expression telling her. Anyway, so that was it. And then the whole audience got a nice laugh. So you see personality traits already when you are 5 or 6 years old. So here you go. This is my sister today. She's a principal. She's a mathematician. She's a principal in Navadisha Montessori school in Chennai, South India. Actually, her son was with us. He just graduated with mechanical engineering degree from here. You will see him later on. So now coming to grade 10 finished. This is the board exam, and you have to go into college, and we are impatiently waiting for the results. The results were, of course, not online at that time, so there is a delay before you get the results in the local school, and the local school actually announces the results. So I was not anxious. I knew I will get enough marks to get into college, but I was impatient. So I started putting some pressure on my father. I started talking to him. You should know somebody in the head office, so you can maybe talk to someone, and maybe once the results are finalized there, we should be able to know how I did. My father said, I do not know anybody there. Even if I did know somebody there, I was not going to pull any strings, and you are going to wait until your school, just like every other student, your school declares the results. I started my drama. I remember I was not talking to him for a week. I avoided sitting for dinner when he was around. He kept his cool. He knew I will get over it. He's the type of person, he used to read the books like this. I think most of you in the audience know Mahatma Gandhi, father of the nation, and it was actually his leadership and non-violent freedom movement that got independence to India in 1947. No single bloodshed. That was how he was. Not intimidated, not anything. So my father was of that level of thinking and peace, and as I said, firm when they needed to be. And this is his autobiography, The Story of My Experiments with Food. Beautiful book to read. I have read it once, I should read it again. So anyway, the long story short, my drama did not work. So I had to wait for the results to come out in the local school. I remember the day till today, walking to the school to find out the results. This is my friend Usha. She used to score slightly more marks Madi in every single exam. So she came first, I came second. So there was competition. Well, many of you know what competition means. So, and I was walking to the headmistress' office, and I meet one of the teachers. So he asked me, "What's your name?" I said my name. He said, "I think you are top in the school." My heart started beating. I'm like, "Are you?" Anyway, we walked to the headmistress' office, and we verified the results. So this was the result. I couldn't believe it. In fact, we both were top in the school. How can there be a difference of 1 mark? It was just bad luck for her. Luckily, I did not have to face her on that day, so I got saved. So this is my school. This is my teachers and the friends. Some 15 or 17 of us. Some couple of them are no more. My chemistry teacher is unfortunately not in this photo. She was not there on the day. Anyway, so after class 10, grade 10, my nephew told me, "You used the word grade 10, not class 10. So I'm just, trying to practice. Anyway, so I went to college, pre-degree, so 2 years. And after pre-degree, what are the options? Of course, you can go into BSc, but also these are some of the other popular options. People go to do the bank test. So if you crack the bank test, and if you get a job in a national bank, doesn't matter whether you have that degree or not, you may do a degree later on, your life is set. Because you are in a national bank, you get promotion, that's fine. But your math need to be very strong, which was not in my case. So I did not crack the bank test. Equally popular, in fact, more popular, the engineering and medicine. So again, as you know, there are a lot of people, but there are a lot of smart and bright people. So if you want to be amongst them, and if you want to do well, you can imagine to getting into any of the schools, the competition we face, right? You have to be better than the best amongst the millions. So again, I didn't get into engineering or medicine. So the next best thing to doing engineering or medicine was, of course, to study chemistry. So I studied BSc chemistry in the local college, and 3 years. And after that, what do? Of course, I wanted to do MSc, but then again, there was a small hurdle. If I remember correctly, in Kerala, the state of Kerala at that time only had, I think, 20 seats for MSc chemistry. And that's because, as everybody knows, chemistry, like every other science discipline, is a laboratory-based science. So to get accreditation for a college, you need to have proper setup lab. So only 20 seats. So if you are 21st in the state, you are out. And I didn't plan well. I didn't apply anywhere else outside of the state. So I had a little bit of time to wander around. So that's what happened. And I was actually wondering, while also actually wandering into doing English MA literature. So I did that for 1 year. I knew that this is not what I'm going to do. So the following year, I was more prepared, applied for MSc to many universities, and I got into one of the top national universities. This is very far from home, up north, Banaras Hindu University. So actually, I visited BHU just before COVID, and this is our organic chemistry lab. This is a photo taken in 2020. It was a very nice moment. So that's our chemistry department, and this is our MSc class, and this is one of my professors. So there I was. Now, when I reflect on this, I think it was the right thing. It was a good thing for my career. If I had studied MSc in a local college, just a college, I perhaps would not have gone ahead with my PhD and so on. I don't know, right? We never know. But this is a national university, beautiful campus, everything there. So you get a different exposure when you go to such a university other than studying in a local college. So that was opening the door for my future career. I got a small scholarship to do PhD. This is from the University Grants Commission. So this is their logo. That is the Ashoka Chakra. Chakra means wheel. You can see the wheel there, and there are 24 spokes in that wheel. And the 24 spokes essentially signify the principles of Lord Buddha. And then we have the flying bird, and then we have the open book. The three things together essentially again signifies the importance of knowledge and how knowledge can be liberating. So that was always in our minds, and in the family, the bar was already set high. My uncles, my aunts were all doctors, engineers, policymakers, and so on. So the bar was already set high, and there was no escape from not going to higher education and studying well. So many of you, at least some of you, the biochemists will know this. This is Professor G.N. Ramachandran, first to propose the triple helical structure of collagen. In fact, this is the first structure of any protein molecule available in the literature. So he was actually the student of C.V. Raman, the Nobel laureate. Everybody knows, at least the scientists in the audience know. So he went ahead and looking at the fundamental structure of peptides and proteins. And that is the Ramachandran map. Again, all the biochemists in the audience know this is a Ramachandran map, and this is now part of every single textbook around the globe. Biochemistry students study this. This is essentially translating a three-dimensional problem into 2 dimensions. I won't go into the details, but it's essentially telling us what are the allowed conformations a polypeptide can take. Now you have to remember, this was done in the 1950s. No supercomputing facility, nothing. It was out of the brilliance of his mind. On a piece of pen and paper, he deduced all these calculations, and later on, when the protein crystal structure data became available, the two perfectly match. The theoretical calculations perfectly match the practical findings. So Molecular Biophysics Unit, Indian Institute of Science, Bangalore, that's what he set up in the 1970s, so that's where we worked. These are my PhD mentors, Professor R. Balaji Rao from BHU and Professor P. Balaram from Indian Institute of Science, Bangalore. Very simple, humble people, no luxuries, no nothing. Professor Balaram sits in a corner of the lab, you will see him reading. Every single Sunday, you go to the library, Professor Balaram is there, he puts his son to sleep, and then he comes to the library. He is reading Jacques, Angewanda, the chemists in the audience know nature, science, and so on. Everything actually at the tip of his tongue. And he's also a very eloquent speaker. So we all try to sort of emulate him, but we fail. So actually, I do that. During COVID, everything locked down, so there's all of these podcasts and interviews, right? A lot of his these talks are available, and I still watch it, and we try to learn from him. And also a lot of the history behind the science during that time, that's where I pick some of these things from. So I did postdoc, as you heard, in Sweden, where I worked on 4 helix bundles. This was Linköping University, Professor Lars Balser. Leeds, I was there for a year and a half. Professor John Collier worked on some larger proteins. Texas A&M University, I worked a little bit on TLR3 protein, and that was with Professor Cheng Kao. So before we proceed, let us have a look. For those of you in the audience who do not know what peptides are, I was trying to talk to some non-scientists, and they said, what are peptides? So these are peptides. Peptides are essentially smaller proteins. Proteins, everybody knows, well, the large molecules. And you these days hear a lot of, you can get a lot of these peptides available in the market for muscle building, skin elasticity, and so on. This is another interesting molecule. This is it's a dipeptide. It's a low-calorie artificial sweetener, 200 times sweetener than sucrose. It is FDA approved for use in food. So now let's look at what are peptides made up of. Peptides are made up of these molecules called amino acids. I have color-coded the amino group and the carboxylic acid group here. There are 20 protein amino acids. That is the amino acids that form the building blocks of the mammalian proteins. There are other amino acids in lower organisms, but this is the mammalian proteins. They have abbreviations, letters, different letters. I have also color-coded the amino acids. That's essentially to indicate that there are some differences between them in their nature, particularly when you try to incorporate them in proteins or peptides. The black ones, well, all I would say is that, so they have side chains and so on. They have slightly more number of carbon atoms on the side chain. The other colors, they would have more number of nitrogen and or oxygen atoms as part of their side chain. So now we have 20 letters sitting here. So we can essentially combine them together and make peptides. And some of them could be meaningful words, as you can think of. So let's see what they are. Here you go. Chemistry is a peptide, if you look at it, right? It's a peptide made up of these amino acids. Actually, don't worry, Jan. Physics is also a peptide. No need to get disheartened. Physics is a peptide. Sorry for the biologists. Biology is actually not a peptide if I go by the table here. Anyway, sorry about that. Now let us look at, well, today's speaker is Viji Sarojini. They told you that she is a peptide chemist. Now we are going to do a fun exercise here. Is Viji a peptide? You have to look at the amino acid table. Fortunately or unfortunately, Viji is not a peptide, because there is no single letter with the amino acid with the single letter J. So Viji is a peptide chemist, but she is not a peptide. How about her husband? He is a peptide. Look at that. In fact, if you look at the amino acid composition here, there's a balanced one. Well, a more or less balanced, I would say. Okay? So more or less balanced. And now, allow me to say that many people in the audience are peptides. You can verify for yourself whether you are a peptide or not. Now, if you are a peptide, I am going to come and study you, because that's what I study. So there will be some investigations, because that's what our group is interested in. So all the peptides in the audience, just watch out. Okay, so now let's move on. And here I have shown on the right-hand side what we call non-protein amino acids. So these are slightly different to the ones you saw on the left-hand side, slightly different in structure, and we use them. These are the amino acids which I used and some of my students used. So we'll come to that soon. Some major milestones, well, the biggest is, of course, insulin, the discovery of insulin, 1921, and 1922 it was published, and 1923 was Nobel Prize. Now you see here, when the Nobel Prize was announced, the contentious relationship, it seems, that existed between these two people aggravated. Well, we are all human, aren't we? You know, there is competition, there is anti-competitive behavior, jealousy, and so on. This is 100 years ago, more than 100 years ago, and these are Nobel laureates, but they are human. Second Nobel Prize for insulin in 1958. So now let's look quickly at proteins and the protein structure, hierarchy in protein structure. Let's see, these are the amino acids. We saw them before. So they form these structures. These are the proteins. They are large and complex molecules. As you can see, different shapes, different turns, twisting, and so on. And that you see here is the tertiary structure, and that's the functional form of a protein. We as chemists cannot actually synthesize these proteins. They are very large and complex molecules. So we as chemists, we settle down to the middle here, and these are peptides, right? So these are smaller proteins. So they adopt different secondary structures. As you can see, they can be a helix, be it a strand, be it a hairpin, and so on. You will see some of them, what we did in the lab later on. So we can essentially mimic these structures, we can make peptides, we can think about introducing function, and so on. An important point here, if there is any single mistake, well, I use the word mistake in this audience, but it's called as mutation in science. If there is one single mistake here, it can actually be fatal, or it can be very serious diseases or disabilities. Now I have to share a very sad story that actually happened. This is called as the sudden death syndrome. It seems babies just die. Babies like 2-month-old, 10-year-old, 5-year-old, 3-year-old, they just died. There was no particular explanation for why these babies were dying. And this used to happen. There was not much knowledge about it. And this family here, their life turned upside down just because of this. This happened in Australia. This was maybe 25 years ago. 4 children, one after the other, died. When this kept happening, eyebrows were raised. The mother was tried in a court of law. She kept saying, I did not do anything. But circumstantial evidence, whatever that is, she went to jail. She spent 20 years in jail. And all the 4 children were dead anyway, and the mother in jail. Later on, someone decided to reinvestigate using scientific evidence. It turned out that 3 of the babies had one single, I will use the word mistake, but it's a mutation in a protein, and that protein is calmodulin. Calmodulin, proper functioning, is crucial for the functioning of our hearts, especially in younger children. And 3 babies carried this one single mutation, and they died. Based on scientific evidence, after 20 years spending in jail, she was released. If at all we had this data available 25, 30 years ago, at least she would not have gone to jail. She would not have had the agony of losing her kids and spending 20 years in jail. So there you go, the power of science. If we have that knowledge, and how we can put that for human use. So this is actually really a touching story. And so we have to make the observation in the lab, of course, and that is the knowledge we develop. Not necessary that whatever we do in a university setting, a research lab like ours, may become a product in the market. But that is not hugely important. It is important, but it's not hugely important. What is important is the knowledge we generate, and for that we have to make these observations. And because everything has to be explainable, that's what I tell my students, right? Things did not work, that's okay, but we have to find out why it did not work. Perhaps I made a mistake, perhaps I added the wrong reagent, that's fine, but as long as things are recorded and we have, it has to be evidence, it's not magic. So that's what this tells us. So how do we make peptides? We combine the amino acids. By now you would have understood 2 ways. Solution phase and or solid phase. Now it's like you make the chicken as a curry, which is a little bit messy, lot of cleanup and so on, or you can get your meal ready by putting the chicken in the oven, and that's the solid phase. At least that's my analogy here. So it's very easy when you do the solid phase. Well, I wouldn't say very easy, but much easier than the solution phase, especially if you are making a long peptide. So you can essentially, clean up is easy, you just throw that aluminum foil, which is the resin, and you get your peptide. So why do we use non-protein amino acids? That's because peptides made up of the protein amino acids can adopt a whole different type of structures, shapes, I would say. They're very flexible. They break down in stomach, and so they are not very useful for many applications. And as many of you may know, most of the peptide drugs have to be delivered by injection because they degrade in stomach. So that's why we use non-protein amino acids. We get better control over what sort of shape your product peptide may adopt. So that's the reason. So for my PhD, I was actually synthesizing these non-protein amino acids. You can see some hazardous reagents we are handling here, potassium cyanide. And also, the chemists in the audience know the Haydn-Toyn reaction. You have to generate the anion and so on. I won't go into the details. But it's a very sensitive reaction. So you have to carry that out under inert atmosphere, liquid nitrogen minus 72 degrees Celsius and so on. So I have to acknowledge my senior here, Dr. S.K. Misra. He helped me a lot to set up these reactions, particularly to get large containers of liquid nitrogen. You know how it is. Very nice man, unassuming. Unfortunately, he is no more. So these are the non-protein amino acids I synthesized as part of my PhD. You can see them. Now, here I have shown 2 peptide-based drugs that are marketed. This one here, semaglutide, available as an injection against type 2 diabetes. Later on, people changed the sequence of it slightly. They incorporated AIB, and this was a turning the tide moment, because now semaglutide is actually available as a tablet. So that's an example of the importance of using non-protein amino acids. Some basic work from my PhD, so lots of secondary structures, NMR, CD, and so on. I won't go into the details, and these are difficult to synthesize because we have these non-protein amino acids. They are bulky, so incorporating them in peptides and then cleaning up and looking at the structure. A lot of hard work. I used to synthesize 3 to 14 residue peptides and study them by NMR, all of them published. For postdoctoral, I worked on slightly larger polypeptides, also putting other functional groups, that's like a sugar into the peptide, or other functional groups. Here we are essentially trying to emulate the proteins like enzymes. So we put different functional groups and see if they can come together and catalyze a reaction and so on. So this is another example, what we call 4 helix bundles. We introduce some negative design and then bring 2 different peptides together. They dimerize, so we call it heterodimers. We got some unexpected results as well. So down south it is now, because we were in the US, and I actually got a job at San Antonio, Texas, in one of the medical facilities. My husband was just finishing his PhD. I was, to be honest, not that keen to continue living in the US. I said that work-life balance is not good, etc. I'm not sure if it's good here, but it definitely, that was my opinion at that point in time. In any way, my husband, he got a faculty position at the University of Auckland, engineering. So we moved here. I was looking for a job, so I got a 1-year contract at Plant and Food Research. So I worked there. That's where my interest in plant protection started. So we published a paper, a novel structure, and the following year, I did not have a job. So actually, there was a job offer in Wellington. This was IRL. So this was a research position, and I finally decided not to go, the 2-body problem. My husband was already here. And at that time, the Dean of Engineering was Professor Peter Brothers. And so he had this, he would welcome new staff and talk to them about their family and so on. So he was asking my husband, and when he said, my wife is a chemist, so he suggested that maybe you should ask your wife to go and at least see my sister. She's in chemistry. So thank you, Penny. So Penny is the first point of contact I had in chemistry. I was working at Plant and Food at that time. I remember this was the day of the inauguration of the NMR Center. 2005 should be. I think Brent knows, he's smiling. So I came here, and this was the old building, and I talked to Penny, and then I think my CV was floating around looking for jobs. And the following year, a teaching position in medicinal chemistry became available. I decided to accept that job, and my job was to teach in medicinal chemistry, these undergraduate courses. So that was a big change for me, because I came from a research-only background, and now I had to stand in front of a large class like this, and I was talking to students, Kim Wan-Chen, and so on. So, however, because I was interested in continuing my research, I continued that, and there were no this thing, so I continued that in my spare time. Here I have to thank my great colleagues here, Katrina, Sandra, and Jeff. So they would actually very happily lend me a fume hood, thank you all, and also some small equipments, and that's what I was doing. So I would synthesize non-protein amino acids, make peptides all by myself. And okay, I got a new staff research fund. I think this was 2007, $13,000. So research was slightly picking up, and the rest is history. So here you go. So this is my first MSc student. So she was designing this beta hairpin structures. And later on, when Alan joined, he continued that work. And you saw this amino acid. Some of you may remember, non-protein amino acid. We incorporated in an antibacterial peptide, published it in 2017. Kyriakos continued the work, lots of beautiful structures. Alan was interested in cyclic tetrapeptides because they are difficult to synthesize. And he said he wanted to do that. And the course of his investigations actually led to us discovering a beta turn here. This is you saw the word beta turn before. It's a different type of beta turn. It has hydrogen bonds. It is within the definition, but it's not the one that's found in proteins. Beautiful work, all published. We got so much interested in cyclic tetrapeptides, we decided to write a comprehensive review article, and this is actually a very challenging task. Chemists in the audience know if you want to publish a review article in Chemical Reviews, well, I will never do it again. So this was a formidable task, but we decided to do it, and it got finally published. And this is my close friend and ex-MSc classmate, Professor Sanjayan, back in India, National Chemical Laboratory, Pune, India. So the most intuitive organic chemist I have ever talked to in my entire life. He dreams organic chemistry. That's his dream. So that's Sanjayan, and the people in the University of Auckland people know that's distinguished Professor Bill Denny. So we co-authored the Chemical Reviews article. Antimicrobial peptides is a major part of the research that goes on in my lab. These can be natural products. As you can see, these are the sources. We produce antimicrobial peptides as part of the immunity. We as chemists can also design things de novo, do modifications, and so on. This is a molecule, colistin. This molecule was known for more than, I think, 70 years. It's a natural product antimicrobial peptide. It's produced by soil bacteria. It was used and then it was withdrawn from the market because it has some dose-dependent toxicity to kidney organoids. Later on, it was reintroduced into the market because there are no drugs that can actually treat certain gram-negative bacterial infections. So colistin is still there. This is a very close analog of that molecule, colistin. And this is my first PhD student, Dr. Heru De Souza. He is working at Douglas Pharmaceuticals now. So this is a cyclic peptide, again, very similar in structure. He did extensive work, lot of peptide synthesized, looked at the activity, finally found one lead peptide, and this seems to be much well tolerated by kidney organoids. That part of the work is not yet published, but the previous work is. Well, everybody knows the word resistance, right? We all resist things, right? So we resist, I don't know, family, friends, whatever. So do bacteria. So they resist. So that is antibiotic resistance. We throw antibiotics at them. After a period of time, they're going to tell us, you know what, I am not going to be intimidated because your antibiotic is not going to work. So that's what happens. And that's called as biofilm development. Biofilms can grow on surfaces, especially medical implants, major problem during surgical procedures and so on. Once the biofilms forms, you have to surgically remove it. The biofilm work started in collaboration with Professor Gillian Lewis and Vidya Washington in biology. Later on, we got some funding from Auckland Medical Research Foundation. So the biofilms, they would have also dead cells and so on. It's a very messy situation. So this fund was essentially to look at self-cleaning antimicrobial surfaces. This is my postdoc, Dr. Delsa Joseph, who was working on this. We have not published this yet. So in the meantime, Heru was looking at attaching these to surfaces. There are some technical terms here, chemoselective immobilization. In simple terms, what that actually means is we are actually going to paint that wall with a mold-free paint so that mold doesn't grow. Mold, in this case, bacteria. The paint is our peptide, and the process of painting is the chemistry we are doing. So that was done by Heru. Brilliant work. The entire credit goes to him, not to me. And there are some color coding here. That is essentially to tell you that, so we don't want everything reacting with everything else. We want selectivity. So the color means that certain things react with certain other things. So we got some beautiful work here. So the bacteria do not grow when Heru puts his peptides on these medical implant surfaces. Again, published in 2017. We are now going to click everything together. So that is the Nobel Prize winning chemistry 2022. So this is a versatile reaction and the clean products, good yield, and so on. That's why he won the Nobel Prize. So my another PhD student, Saurabh, who is here in the audience, he decided to expand this chemistry. So you need to have complementary hooks so that you can hook things together. Click things together. So Saurabh designed a template which has got multiple hooks, so you can hook multiple things to it. You can hook the same peptide or different peptides. So we got lots of interesting results. So these are the hooks, the red ones in reagent 1. The blue is the complementary hook in reagent 2. You can make the peptide brushes, and just one simple result here, the peptide seems to be much well tolerated in serum. So I told you protein amino acids, if you use, the stability is lost and they degrade. So here is an interesting result. We're now going to stick things together, and that is the muscle chemistry. The marine organisms, they have a protein in their food. So especially for the muscle, it's called as the muscle food protein, and this is the chemistry they do. So they have, this is a glue, and it works underwater, and it sustains the water currents, underwater currents. It's that strong. So that's the molecule dopa here, and some other amino acids like lysine, they cross-link and they form a beautiful glue. So that's what Saurabh did again. You form the polydopamine on urinary catheters and then attach the peptide, and we get, see this thing. Another analogy is the Velcro chemistry. So this was it seems this was a Swiss electrical engineer walking in the woods. These sorts of plants, they stick to your garment, and he made an observation. Again, it's about making observations, right? He took that plant to his workplace, looked at it under the microscope, and saw, oh, these have these hooks, and perhaps I can make use of these, and that is the start of the Velcro business, which is a booming business. It seems even NASA uses these types of things from Velcro, so they are very happy. So far, I was talking about, hope I'm going okay in time, I don't know, antimicrobial peptides, and they don't have a specific target. They work on the bacterial membrane. What if your drug molecule has a target that is inside of the cell? You need the drug molecule to reach the inside of the cell. So that is drug delivery, a big aspect of medicinal chemistry and pharmacy research. So this is my PhD student, Kamal Patel. He is currently a postdoc at Harvard Medical School. So the concept originated, the molecule is cell-penetrating peptides, first discovered in the human immunodeficiency virus. That's not the virus uses to infect the host, but it has these CPPs. So Kamal had these peptides. They are antifungal as well as anti-cancer, and that's because there is the same target in cancer cells and fungal cells. These are big proteins. It's called as proteasomes. The struggle is, the challenge is to get the peptide inside the cell because the target is inside the cell. So here we have to do a delivery technique here. So you have your truck, which is the CPP, and you have a chain, that's the linker. It's a 3 amino acid residue, and he packages it nicely, and then it's just like delivering an important piece of parcel. And it goes to the inside of the cell, and it gets delivered. And we got some beautiful results on fungal biofilms and also against cancer cells. So a couple of more concepts here. So this is called as self-assembly. So this is essentially like you stack a lot of coins together. And that's what is happening here. And in order for that, at a molecular level, in order for that to happen, we need these red colored bits in the molecule. That's when the stacking will actually stay. And when these molecules stack together, they will also pick up some water molecules. And then you get the hydrogel. So Hugh developed this hydrogel, antimicrobial, and here you can see when he uses these hydrogels and he tries to grow the bacterial cells and the human cells together, the bacteria die and the human cells survive. So that was a very good result. Saurabh and another student, Aljo, they worked on this concept of self-assembly peptides and used this as a template for other things. such as mimicking artificial photosynthesis. Here we have a porphyrin, or putting other chromophores and putting that metal nanoparticles, trying to do carbon dioxide reduction and so on. So another piece of work. So this is the work in the food space which we have been doing. Well, frost damage is a thing that occurs in nature. That's through ice nucleating proteins, and this is a major problem. For us, maybe it's more of a problem in this space, that is freeze-thaw damage in food. When you, especially cellular food, they lose the structure and texture, and consumer acceptance goes, and also nutrients. So nature provides its own solution to its own problems. So here we go. These are called as anti-freeze proteins, and these are present in polar organisms. So this is how it works. In the interest of time, I won't go into the details. So our interest was to make these antifreeze proteins, or at least the peptides. The issue here is, so I have put the red boxes here, and the word is disulfide, very difficult to form in that order. And even if you form, it's difficult to maintain in that order. So it's a challenging thing for anybody to do. So my another PhD student here, he looked at the sequence and designed a much simpler sequence based on the original protein, and made this antifreeze peptide. To be honest, our antifreeze peptide does not look like this. This is the image I generated using our dearest Copilot. This is what it gave. But it's not a helical peptide in our case. In any case, simplified peptide, less number of disulfides. We can also replace the disulfides with something else. Beautiful results tried on range of food, and we found some interesting activity. You can see very thin crystals form. This was done in collaboration with initial discussions with Conrad. Clive Evans, some of you may know, taught Charles how to do these measurements and so on, and the NMR in collaboration with Ivan. Plant diseases was always an interest to my group, and we have done a lot of work in this space. More recently, Rizana, she just submitted her thesis, fungal diseases of plants, strawberry, grapes, and so on. This is from her result. When she puts her peptide, it actually works beautifully. And those of you who take walks in the parks know that many, especially Vitagrey ranges and all, the parks are, at least parts of the parks are closed for public. And that is essentially to minimize the spread of the cowrie dieback and other diseases which caused by this organism called Phytophthora. So there is a lot of interest in doing this. The problem is the different stages in the life cycle, and these are soil pathogens. They infect the roots and keep infecting new hosts. So this is a new zoospore video. Zoospores move in soil, and that's how they do this. And this is it's very difficult to grow them. So I just got this from Chris. He was very happy last week because he got these zoospores working. And so our idea is to use our peptides and natural products from New Zealand native plants, and to work on some solutions for Phytophthora. So here I have to acknowledge Michael's statement. Our initial discussions with Michael was instrumental to start our work on horopito. Buligodiol is a bioactive. So that's what we are working on. So a lot of work went into this. Heru was part of this work, and we published a paper in 2023. So we wrote some funding applications. Surprisingly, we got the fund. It's called as Smart Ideas. And you can see a lot of people involved, plant and food collaborators. They're all here. Thank you for coming. So what we are trying to do is bait the pathogens in the soil. And this is from Heru's results we published. Currently, 2 of the other PhD students are continuing this work. Our molecules don't seem to be harmful to the cowrie leaves. The project is essentially to do these hybrid drugs. This is again a concept in medicinal chemistry. I won't go into the details. It's like the Lego chemistry, right? So you have the different pieces and you are putting them together to get some meaningful structure. But for us, function is also important. And that's being done by Ishita. She's in the audience. She's synthesizing these hybrid drugs. So that's all the research. So now quickly going through some of the teaching, if I have a few more minutes. So as I said, I was teaching in all of these undergraduate courses and some postgraduate courses. At that time, we did not have the Fisher and Paykel lecture theater. So Chem 110, we used to give lectures. This is large chemistry. You can see large chemistry and medium chemistry here. I promise you nobody was drinking before going into the lectures. This is just an old photo I found in one of our shared drives. And actually, that's our ex-dean, Professor Grant Guilford. So this was the photo I could find. There are no other photos of large and medium chemistry anywhere. This is our great colleague, Dr. Judy Britton. She was the director of Chem 110. We used to give this lecture 3 times a day, 8 o'clock in the morning, and then 11 o'clock, and then 2 o'clock. So by 2 o'clock, your sound is totally gone. I used to coordinate this other course and industry and so on. That was then, this is today. So this is Chem 351, a new course which we developed as part of our undergraduate chemistry curriculum. And here, actually, we are going to make a lot of money because we are making silk. So you look at that. So actually, the students are not synthesizing silk. They are synthesizing a peptide fragment based on silk that will have the structure, the nanostructure, similar to silk. So that's what we are doing. These are the GTAs who helped me run the lab. That's Ishita, and this is John here. So when I started working here, so Chem 390 is a core course for medicinal chemistry. So we used to synthesize this drug molecule. This is sulfathiazole, and you can see that it's antibacterial. At that time, we had the old teaching labs, no fume hoods. So students used to actually do this reaction, reflexing it on the bench. We used to use some nasty solvents, very nasty solvents, pyridine. Chemists in the audience know. So I decided to do a little bit of research, and we changed the procedure. So we now do this synthesis following this benign procedures. In fact, this is what our 390 students were doing in the teaching lab 2 weeks back, synthesizing this. And this is a paper co-authored with Jeff and Sandra here. We worked together on this. So almost finishing. I've also been doing this job as the academic head of the school. So the university interviews their staff, and they ask lots of questions. So here is a question. Describe some of your day-to-day research activities. Everyone will say something like this. Research ideas, I talk to my students, develop, and then grant writing, this and that. In reality, well, it is there, but it's not day-to-day. That's because there's a lot of other things we have to do, which is fine. That's part of the job. That's all right. Now, one of the things we have to do is, it's a big university, lots of classes, so we have to set the timetable. We have to set the timetable very well in advance. Actually, we are doing that right now for 2026. If we get it wrong, everybody will lose their jobs. So here we have the timetabling data collection process. So if there are any software people here or computer scientists, I'm going to apologize, because every single year we feed the data into this software. The following year, it comes with totally wrong things. Now, if the university ever decided to verify what we write in our promotion application about teaching, that I told you I'm teaching all these courses, if they decided to verify that with what the timetabling software data says, nobody will tick the box, because none of us are teaching. In fact, Penny, you are teaching. 320, please come back, you are welcome. So you are teaching. And so this is the joy of dealing with the timetabling software. So this is a term I coined. So those of you who work with the timetabling software and the academics perhaps can make some guesses here. Any guesses? So OCTD, it stands for? This is the problem. This is actually a real problem. That's why I said I have to apologize to the computer scientists and the software engineers here. This is what the software does. This actually happens. It's happening right now. My great colleague here, David, and me, we are talking to the staff and saying that, by the way, who is there? Tristan, you're not getting, Jin Young, you're not getting any credit for teaching 351 because you're not teaching, according to this software. That's what's happening. Only I'm teaching. And in fact, David Ware is teaching. David told me, I'm not teaching Viji, which means essentially I am teaching. So that's the timetabling software. Almost there. Lots of professional activities during the years. What is this? The open day here. Actually, it's coming up in August. We're all asked to attend that and contribute. The research showcase, I was chairing and also member of the committee for a long time. The New Zealand Institute of Chemistry, we used to run pub quiz and so on, some nice photos. Center for Green Chemical Science, I've been part of this. A big thank you to all of my students, especially to the PhD students who have done all the work. In blue font are the people who already graduated, and in black font are those who are actually still working. Some nice pictures from here. Actually, they know I'm not a very social person. I just, I cannot, but we still try to do it sometimes. And so this is some nice pictures from those nice moments. A lot of people to thank here in the university, the entire technical services team, particularly Pawan, great colleague, and Rajesh, Tastik, and everyone else. Teaching lab, beautiful colleagues. Again, research program coordinators, we wouldn't do anything without you. Thank you very much. Group services, again, Minyang here. And despite what the timetabling software does, I'm going to thank the timetabling committee, the people who work there, the finance team, each and every academic colleague in the chemistry department. You are great colleagues. Thank you very much. Collaborators in other departments and faculties. You saw many of them in some of the slides, and those are the collaborators. Previous and current heads of schools and deans, acting deans, deputy deans, associate and assistant deans. I work with them a lot in the academic committee, Bruno here, and also Tilo here. And I was also part of the academic programs committee, so spent 3 years working there. So I should thank them. And the School of Graduate Studies, they help us run our exams, PhD exams especially. And these are the people who gave us the money, so thank you to them. Big thank you to a lot of family and friends. So some of whom you saw before, my grandmother. This is my grandmother's sister. And she, none of them, they are no more. Her sons and daughters, my uncles and aunts, great supporters of us establishing and becoming where we are today. So special thank you to them. This is my mother's younger sister. She had some health issues. She passed away long ago, actually suffered a lot. Another aunt here. This is my sister and her husband, my sister-in-law. This is my in-law's family. This is actually her husband's niece, Dia. She and my nephew, they were here. They were both University of Auckland students. Dia is still a student. She's doing medicine here and third year. So we used to have some great, actually during COVID, the whole thing locked down, 4 of us locked inside the house. That was fun times. Anyway, here you go. And this is my nephew. He graduated with mechanical engineering honors from University of Auckland, currently working in Wellington as a mechanical engineer. And this is a beautiful photo from when we were in Texas A&M University. These are my cousins, and I need to thank all of them. And these are some beautiful photos from our hostel. And this is a photo taken in 2025, February, when I went to actually the Indian Peptide Society meeting. These are 10 of our grade 10 friends. So there was a small get-together. We actually had a great time. I should thank them. And finally, just a piece of dedication and closing remarks. So this is hard. This was my close friend, Kaveri Acharya. She was a professor at Delhi University. She succumbed to motor neuron disease. I would like to dedicate the talk to her. Many of you may remember this incident. This happened like 6 weeks ago. And this, Kyle, a brilliant student, PhD, doing at the University of Auckland. He came from the US. He was in biology, and he was just killed by some random attacker, not in a dodgy suburb. This happened in one of the reputed suburbs right here in Auckland. And this is the suburb where we live, some of our colleagues. This is the bus stop. We actually take the bus from almost every single day. These are eye-opening self-reflections. If you are at the wrong place at the wrong time, it really doesn't matter whether you are in New Zealand or New Delhi or New York or New Caledonia, it is the same thing. there is no discrimination at that time. So that's really humbling to note that, in fact, I was reading some of the interviews for Kyle. His father said Kyle's dream was to become a professor and perhaps give a talk like this, which I am doing today. He didn't make it. Aside of these misfortunes, of course, we know, I definitely know, that there are many other people out there who are smarter than anybody in this room. However, they did not make it, not because they didn't try, not because they didn't have the drive, but circumstances were not favorable. That's essentially what I'm trying to tell myself. We celebrate everything. That is fine. We can be aggressive. We can be ambitious. This is not to say we shouldn't be ambitious, right? We shouldn't be lazy. But while we celebrate all these things and remain ambitious, remain competitive, we have to also remain humble while knowing that, well, I won't say sky is the limit. Sky can be the limit if luck is on your side. With those words, I would like to thank you all very much, and thank you for listening.